So we're coming into the home stretch. We've only got one full week of classes left and a little bit week after that. As much fun as this semester's been, I know I'm looking forward to it coming to an end. And I imagine you are too. But remember what we talked about early on when we first talked about the classes. This is something that you're going to remember for the rest of your lives, and you're going to be telling your children and your grandchildren sometime down the line about the pandemic of 2020 and how you came through it. And so remember that. Remember, it's going to be a story to tell. And speaking of stories to tell, so we're going to be bringing together now and finishing our story about how we got to where we are. And where we're going to start pulling our chemistry together, what we've learned this semester, together with what we've learned about the viruses and DNA and understanding the story of life. We're going to begin to pull those together as well. So our story today has to do with DNA sequencing. That is actually reading the DNA of a species. Now, used to be incredibly expensive and incredibly time consuming. When I say used to be, I mean as little as 40 years ago. So I know to you guys that sounds like a long time, but to me it's not that long. But 40 years ago, the idea of sequencing something quickly was unbelievable. Yet, when the coronavirus was discovered, the Chinese managed to sequence it in a month, perhaps. Sometime early in January of 2020, they had sequenced the DNA sequence or the RNA sequence, same thing, the genetic sequence of the virus, and made that available to the public. Now, understand what that is. We'll look a little bit this next time. It's everything there is about everything there is about the virus is right there. They were able to read it. Whether they understood it or not is a whole different matter, but it was all right there. So the trick that made sequencing possible was this discovery by this biochemist by the name of Kary Mullis. Now, in order to sequence something, in order to study DNA, a specific region, a gene, something like that, you need many copies of that DNA. And how are you going to get these? If you start with one virus, you've got one copy. How are you possibly going to make many millions of copies of this thing in order to now read it? And this was what Kary Mullis was thinking about when he was driving along the highway one night. Now, to understand his thought process, we need to pause a minute and remember what's happening when we heat something up, and in particular what happens when we heat up DNA. So DNA is this double helix. So it's this double helix wound together like this. And when you heat it up and get it hot, basically the helix comes apart into two separate pieces. That's the first thing we need to know. The second thing we need to know is we see the same process with proteins. So a protein, when you heat it up, it unfolds from its shape, from its arrangement where it has some function, and basically loses that function. And we've all seen this when we've cooked eggs. So eggs are mostly protein, and when we heat them up, what happens is they do what we call denaturing. So they denature and they unfold now, and they lose whatever function they have. You can't get it go back the other way. Now, you and I know why this is. It's this way because of the second law of thermodynamics. There's one low energy arrangement of the atoms. There are many high energy arrangements of this. So when we add energy to the system now, it distributes that energy amongst all the possible states. And so we see all of these high energy states now are expressed. So DNA unfolds, unwinds when we heat it up, and proteins lose their function. Now, the other thing you need to know is that every single species has a protein in it called DNA polymerase. And what DNA polymerase does is it basically is part of the process that duplicates a cell, that makes one double strand of DNA into two double-stranded DNAs. The way it works is the DNA polymerase goes along and reads along this thing. It starts at an end and reads along this thing, and as it reads along, it duplicates the DNA and makes it now another helix. So it starts out with one helix and turns it into two. And every species has DNA polymerase. Now, they're not identical. They differ from one species to another, but they're all there because it's part of the machinery of reproduction. Okay, so now with those two pieces of information, let's go back to Kary Mullis. And what he does is he is working for a company called Cedars Corporation. It's 1983. And he synthesizes short strands, short polymers of DNA. They're called oligonucleotides. So these are about 20 long, so it's like a TAGC, some pattern you want, and he can synthesize these things. Now, in his book, he describes the process that he's driving along one night, and he realizes that the big thing he needs to do is, if you want to study DNA, and everybody wants to study DNA and read it and do those things to it, you've got to make mini copies. And so this is, how do you make mini copies? And this is what's going on in his mind as he's driving down the Pacific Coast Highway one night. And he described it as just being, whoa, he's got this idea. So he says, what would happen if I took DNA and now opened it up? So I were to heat it up and make one double strand into two single strands. And says, I know how to synthesize these little oligonucleotides. Supposing I synthesized about 20 of them that bound to specific parts of these two pieces of DNA. So one would go on one piece of the DNA and one would go on the other piece of the DNA. What he realized was DNA polymerase would now come along of its own accord, just give it the right environment, and it would come along and it would synthesize on this one a new piece of DNA to complement this strand, and on this one a new piece to complement this strand. And so he would have started with two pieces of DNA, and now he would have started with one piece of DNA, and now he would have made two double helices. Well, if I can do that, I could heat it up again, break those two double-stranded helices up into four single-stranded pieces. I could add those little sections, which are called primers, that would go on one end of the gene and on the other end of the gene. And now along would come DNA polymerase, and it would do this over again, and it would basically now go from 1 to 2 to 4 to 8 to 16. And I think you can see by the time you've done this process 20 or 30 times, you've got an awful lot of DNA of the exact sequence you want it to look like. And you pick the sequence by synthesizing the right primer to begin and end the process. So by picking the right primer, you now have made millions of copies of the area of the DNA you want to study. Wow, that was cool. Now, he immediately went about the process of trying to figure out if this had ever been done before. This is such an obvious thing. Somebody must have thought about it. So he did all of his due diligence. He went to the library, he talked to friends. Nobody could find any evidence that anybody had ever thought of this. So after a period of a couple months, he gets to the point where he's confident. He says, I'm gonna try this. And he tried to duplicate a piece of DNA and make many copies of it. And he had some problems because the reaction was so slow that it took too long to do. And then when he heated it up, well, that denatured his DNA polymerase. So now I had to take the old DNA polymerase out, put new DNA polymerase in to do it again. This was just a laborious process, and it was too slow. Now, he couldn't run this thing at high temperatures, which would make it go faster, because at high temperatures, his DNA polymerase would cease to function. Aha. Now, the idea was so appealing, though, that they put together a group at the company he was working at. And this group came up with this amazing idea. Now, as I said, every species has DNA polymerase. And in the early 60s, and still to this day, people study what are called extremophiles. These are organisms that live in extreme environments. And the particular one we're interested today in is one that's called Thermus aquaticus. And it's a bacteria that lives and was discovered in the 60s, I think it was the 60s, was discovered in the hot pools in Yellowstone. Now, before they discovered this, it was pretty well accepted that, in fact, bacteria can't live above 55 degrees because what's going to happen? All their proteins are going to denature and they're not going to work. So you can't work above 55 degrees. Now, and that's 55 degrees Celsius. Remember, our body temperature is about 34, 37, something like that Celsius. So 55 is pretty damn hot, but above that can't go. Well, these guys discovered a bacteria, Thermus aquaticus, and it now lives very happily at 70, 80 degrees Celsius. So that means its DNA polymerase must be what they call thermally stable. So its DNA polymerase is thermally stable, and it also works at high temperatures. So, and the name of this molecule, the DNA polymerase, is called TAQ, T-A-Q. And so the group that Kary Mullis worked with said, hey, why don't we use that DNA polymerase from Thermus aquaticus in our reaction? Because now what we'll do is we will run the reaction at temperatures like 60, 70 degrees Celsius, and it'll work fine. And in fact, at that temperature, TAQ adds 150 nucleotides a second. So think of that. In 10 seconds, it'll add 1,500 nucleotides. So if it binds and works its way along, it just is moving along at this incredible rate of speed because of the high temperatures. All right. Now, so we take our DNA, we heat it up, and we divide it into two pieces. Now, we cool it down slightly and let the primers bind to it, and then the DNA polymerase, which is there and never denatures, and it just goes along and makes copies of this. Heat it back up. Everything we've made divides in two, and then we can do the process over and over and over again. We do it 20 times, and it's an absolutely natural thing to build some sort of a little machine that does all of this stuff. So with that as background, now let's see how we can apply that to understand the coronavirus. Now, one of the most important things with the coronavirus was coming up with a test. And as we all know, in the United States, we weren't very successful in coming up with a test kit. But let's see how we make a test kit. Now, as I said, the viral genetic sequence was completed very quickly, early in January. Now, if you're going to look for the virus, what you want to look for is traces of its DNA. And what people decided, what the world decided, was there were two genes that were characteristic just of this virus. So just this virus, that's the only one. So there was no way some other cold virus was going to be it. They had two genes that were unique to this virus. And what they did was they found the beginning of that virus, that gene, and they found the end of the gene. And they had to synthesize primers for those regions. So gene 1 and gene 2, they found these primers and they put them in there. Now, once they have the primers for these, they can now run the polymerase chain reaction, the PCR reaction. So they take a swab from your nose or your throat or wherever they take swabs from for the coronavirus, and now they've got a little bit of genetic sequence on it. They put it in a test tube, and they now heat it up. They isolate any genetic material that's in there through various ways, and then they heat it up and add the primers. The primers now, if the viral genes are there, the primers will go to that place, glob on, and now along comes we run PCR, along this comes, and it makes a copy of this thing. Now we just repeat it over and over and over again, and pretty soon we've got a whole bunch of DNA that corresponds to this gene 1. If the viral gene was not there, then we've got nothing. We just ran this reaction a whole bunch of times and nothing happened, so we've got nothing. Now, what you can do is there are multiple different, basically, fluorescent stains that you add these fluorescent stains and they bind, they fluoresce. So I put them under ultraviolet light and they fluoresce one color. But they change their color when they bind to DNA. So they might fluoresce, if they're not bound to DNA, they might fluoresce blue. If they're bound to DNA, they might fluoresce orange. So now you take your samples and you add one of these fluorescent molecules to it. The fluorescent molecule goes in and it binds, if the DNA is present, it binds to it. And now you shine ultraviolet light on it, you go, whoa! I have orange right there. So that means that one little DNA sequence I started out with, that one little genetic sequence I started out with, now was replicated millions of times. So there was virus present in your nose or on the swab or whatever it happened to be. So this is so cool. So instantly, within weeks, within months of the time that they discovered the coronavirus, they knew how to make tests. Now, the people at the CDC, they did something kind of different than everybody else, and they checked for 3 genes instead of 2 genes. And one of the tests got contaminated. So there was something in there, some trace amount of RNA or DNA in the samples in the kits they produced, so that it was already present when they checked for this 3rd gene. And so what happens is they were getting all these false positives. They'd add it to water, whatever it happens. But the point is, look at this amazing thing that 20 years ago, the idea of being able to sequence a virus in a matter of weeks and then create a test to check for it and understand how to make that test would have been impossible. And now it's like, boom, and it's done. And if you don't screw up, it works. And so this is amazing. Anyhow, so we're going to pick up there on our next house calls and take it easy, and I'll see you in lecture in just a few minutes.